All right, everyone, can you hear me? Awesome. So thanks for being here for session 8, Sensing and Learning. And I'd like to introduce our first invited speaker, Professor Betty Hong.
Thank you. Good afternoon, everyone. Thanks for coming. And I'd like to thank the organizers for putting together such a great program and for inviting me to be a part of this. So today I'd like to tell you a little bit about the work my lab's been doing in olfaction, our sense of smell. And I've been drawn to this problem for a number of reasons. First among them being that although as humans we move through the world, predominantly our experience is dominated by sight and sound, for the vast majority of organisms on the planet, they operate in the world using chemical communication, using air and waterborne signals that mediate the behaviors and functions that are important for their survival, which include things like finding food, finding mates, and avoiding danger. I might switch to using this one. It's a little brighter. OK, so the other reason I've always been really interested in olfaction is because olfaction or chemical sensing was likely the first sensory modality to evolve. It's the primitive sensory modality, and so offers the possibility of discovering core principles by which sensation and perception are generated in the brain. Now, despite the centrality of olfaction for life on this planet, it remains among basic principles that govern how olfaction work remain hard to glean, and in part that's because of the complexity of the olfactory stimulus space, right? Which, unlike light and sound, are not organized around obvious physical axes such as wavelength or frequency. And the protein-based odorant receptors that detect odor do not themselves provide simplifying principles because they themselves are large, diverse families of proteins that can number in the dozens to over thousands in some organisms. Nevertheless, right, odorant receptors recognize odors based on their structure. And so it makes sense that the search for so-called olfactory primaries, or properties of odors along which odors vary systematically in a manner that would predict their perception, has focused on the physical chemical features of odors. Things such as like their hydrophobicity, their functional groups, steric electronic properties, et cetera. In particular, in the last 10 years, given larger and larger data sets of human perceptual data, combined with new machine learning approaches, we've made tremendous progress in assigning and predicting how structure will relate to perception. Nevertheless, it's been known for many decades that direct relationships between a structure or a feature of a chemical and its perception is the exception rather than the rule. And it's been quite well understood that there are these strong discontinuities in the mapping of structure to perception, such that very small changes in structure in some parts of the odor space can suddenly lead to a dramatic shift in perception. So we know that similar chemicals that differ in just by a few atoms can smell quite different, whereas very divergent, structurally dissimilar molecules can have a convergent perception. So in the last couple of years, my lab has been interested in understanding what might be other additional properties of odors that might be relevant to this question of how odors relate to their perception and behavior. In particular, we've been focused on how odors are organized in natural environments. So if you are a hungry fruit fly flying through this orchard here, you encounter odors not randomly, but in a highly structured manner. So odors are highly structured in space and time. And these are really interesting properties of odors that we're not going to touch on so much today. But in addition, odors are also highly structured in the identity domain. So the natural odors, as they occur in the world, occur as complex mixtures of dozens to hundreds of monomolecular compounds. And the specific set of monomolecular volatiles that come off of this ripe, delicious apple on the branch or this spoiling, rotting apple on the ground gives information about the type and source of that odor, which is, after all, the problem that the olfactory system is trying to solve. Right? So if we look at any data set of natural odors from the world, so in this particular case, this is the volatile headspace composition of a large number of human food sources, where you have odors as rows and sources as columns, you can see that although many odors are present quite sparsely, there are also many odors that are present in specific combinations, and that these correlations in odors across these different sources give information about the type of source. And so what creates this structure in the natural odor space? Well, there are a set of conserved biochemical pathways that are functioning in the natural plant sources that are the source of many of the odors that animals care about, as well as the associated commensal microbes, things like yeast and bacteria, that are shaping the sets of volatiles and their amounts that are emergent from these sources. So one particular, as an example, biochemical pathway that's of particular importance to the organism that we study, the fruit fly, is sugar fermentation in brewer's yeast, right? So Drosophila are essentially yeast feeders, and this is why they are seeking out fermented fruit as their food source. And this pathway generates a specific characteristic combination of volatiles, including acetic acid, acetaldehyde, carbon dioxide, ethanol, and others that create that characteristic bouquet that we can all identify readily as fermentation. Okay, so we have been studying this problem, asking whether the structure of natural odor space is reflected in the olfactory code using this organism, Drosophila melanogaster. And I think that a number of beautiful talks before me have already kind of introduced this model and introduced its many advantages. For us, in particular, olfactory circuits in the fly are quite compact. They have 50 major coding channels as opposed to over 1,000 in rodents. Their olfactory architecture is highly conserved organizationally with that of vertebrates, as well as many of the useful experimental tools, a fully mapped synaptic connectome, genetic tools, as well as in vivo physiology that make studying this system quite advantageous. And among metazoan organisms, the Drosophila olfactory system is among the best characterized in terms of the mapping of odors to receptors to neuron types in the brain. So this is the circuit. All olfactory receptor neurons that express the same odorant receptor project to a common compartment in the brain called a glomerulus. And second-order neurons that are going to read out these signals and send them deeper into the brain extend their dendrites to a single glomerulus. So the glomerulus defines the basic unit of coding, and it corresponds to the odorant receptor that provides its feed-forward excitation in the system. There's about 50 odorant receptors and correspondingly about 50 glomeruli in this system. So second-order projection neurons send their output to two major downstream third-order areas, the mushroom body and the lateral horn. And the mushroom body is thought to mediate flexible or adaptive olfactory behaviors, whereas the lateral horn is thought to mediate more innate olfactory behaviors. And for today, we're going to focus on the mushroom body for a number of reasons that I'm going to describe to you in a second. And roughly speaking, the mushroom body can be conceptualized as a behavioral classifier. So what the circuit is doing is taking the specific pattern of odor-evoked activity among its 2,000 principal neurons and separating them and assigning those patterns to particular readout neurons. So it's a numerically compact version of a cerebellum-like circuit that was beautifully introduced to us yesterday by Martha. What happens is that a relatively small number of inputs, in this case, input from the 50 glomeruli carried by PN axons, is divergently projected onto a large number of principal neurons, these 2,000 Kenyan cells. And each Kenyan cell samples input from approximately 5 or 6, or about 10% of the inputs in this system, and sums these inputs and responds nonlinearly through the spike threshold. Okay? And Kenyan cells are unique in the sense among insect neurons, or at least among Drosophila neurons, in that their connectivity is not fixed and stereotyped from brain to brain. So whereas more or less every other neuron in the fly brain has the same connectivity and the same physiological properties when you record them in every single brain, Kenyan cells this connectivity is probabilistic. So it follows the same connectivity statistics that I just described to you in every organism, but as evidenced by the fact that there are millions of combinations of 5 or 6 glomeruli that you can form out of 50 inputs, and each fly only has 2,000, right? This connectivity is probabilistic in nature. All right, so what this divergent connectivity scheme gets you is a transformation of dense, overlapping, correlated inputs in the periphery, in the antenna lobe and the 50 glomeruli at lower stages of processing, and transforms this into a sparse, selective, decorrelated representation up here in the mushroom body. Okay, and the reason that these inputs here are quite dense and distributed is because each odor activates multiple odorant receptors, and each odorant receptor responds to multiple odors. Okay, so this decorrelation is thought to be quite important for the ability of the mushroom body to efficiently associate different activity patterns with different readouts with minimal synaptic interference between neurons that are part of different representations. Okay, and classical formulations of these cerebellum-like circuits have emphasized the role of random projections or random connectivity in efficiently decorrelating and separating these representations and maximizing the dimensionality of the representation. So, in other words, randomly connected networks would be particularly useful for decorrelating representations in this circuit while maintaining input relationships, which would allow you to generalize between representations, right, that are similar in the input space. So kind of as a cartoony version of this, the idea is that you would push apart representations while maintaining those input relationships. However, it's possible that since the mushroom body is carrying representations that are going to be essentially associated with readouts that are metrics of behavioral meaning or valence, that you might expect that there could be additional sensory transformations that are necessary. In this case, you might expect that odor relationships might become rearranged in the mushroom body, okay? And so these were the kind of alternative hypotheses that we were trying to test as we came into this system. And we did this by imaging directly from the principal neurons of the mushroom body. This is what it looks like physically in the structure. It's why it's called the mushroom body, because it looks like a mushroom. So the principal neuron cell bodies are up here in purple here. The input zone is the calyx that's kind of sitting underneath the cap, and then it projects its outputs through these prominent lobes. So we imaged here where we could assign signals to individual cells. And a very talented graduate student in the lab, Remy Yang, led this study. She expressed a nuclear localized calcium indicator in the stomata of the Kenyan cells, and then volumetrically imaged through this to capture a near complete representation of odor in these cells. And these cells are quite small and densely packed, but she was able to work out methods to reliably segment these across different stimuli. And so she gets population readouts of odors that look like this. So I know it's quite small here, but this is a single trial with time on the x-axis. This trial is 25 seconds. The odor pulse is 2 seconds. And so you can see, indeed, activity is quite sparse as the circuit connectivity predicts. And when we present multiple trials, that we get reliable patterns of activity, right? And we can do this now for a relatively large panel of odors. These odors are chosen to be, in the beginning, we chose these to be physical chemically diverse and to span the input space of the odorant receptors. And we can look at how similar these patterns are to one another by simply looking at the Pearson's correlation of their population activity. And you can see, right, when we correlate every trial against one another and then rearrange according to stimulus, that we trials of the same odor have high reliability along this diagonal. But importantly, on the off diagonal, what we get is a metric of the similarity of the odor representation for those two particular stimuli. So we can see that one hexanol and one pentanol, for instance, are more similar in their representation than to ethylpropionate. All right, so this is what this looks like for one animal. And so we wanted to take a look at what the odor space looks like across multiple individuals. So here is what the representational space in the mushroom body looks like for one animal, for another, for fly 2, fly 3, and fly 4, and we could keep going. And you can see, right, that although connectivity is probabilistic in this circuit, that you get an invariant odor space, an invariant representational space in these flies. And so we can just average together our data across all our individuals, get this mean map of odor relationships. I'm going to reorganize it a little bit so that you can see which odors kind of form these perceptual groups or representational groups in the fly. And then we wanted to compare what this looks like against the comparisons of the mushroom body if it was following these predictions of a randomly connected network. So we implemented a very simple biophysical model of Kenyan cell activity. And this is the work of Matt Bauer, who's actually here when he was an undergraduate in the lab. This is quite straightforward. Kenyan cells were modeled as leaky integrated fire neurons. We can run the model either with inputs uniformly drawn, where every glomerulus has equal probability of being drawn, or weighted based on its frequency of occurrence. And these are tuned to biological response parameters. So we can measure what the input representations of odors look like by imaging in the ORN terminals in the antenna lobe, where they form this characteristic map that allows us to assign signals to individual glomeruli. So these types of data sets look like this. And when we look at relationships in the input space, we can, this is the observed experimental relationship between odors in the input space and in the ORN axon terminals. And we can run those responses through our model and predict what you should see in the mushroom body if you had a random connectivity model, and compare that against what we actually observe. And sorry, I'm going to skip over that for time. And what we see is that what we observe actually deviates pretty significantly from what the predictions of our uniform model are. And in particular, there are some odors that are decorrelated much more than you would expect based on their input relationships. And as well, there are also several instances of odors that are even become more correlated, pushed closer together than is predicted by their input relationships. And these factors aren't changed if we adjust the model for things like slight differences in glomerular numbers in the PN output. Okay, so the view that we're getting was somewhat unexpected, in that what we're seeing is what we think is a reorganization of odor relationships in this mushroom body representational space. And so we went on to try to understand a little bit about what this reorganization would be good for, what would be the functional significance. So what we set about doing is to try to compare different properties of odors against their representational similarity in the mushroom body. So in terms of their chemical relationship, this was relatively straightforward. There's been quite a lot of work in the chemoinformatics domain that allows us to use various metrics to quantify the chemical similarity between odors. But in particular, we were interested in this question of how the relationship of odors in the natural world might relate to their representations in the brain. And this was a more difficult task. And another talented graduate student, Tom O'Connell, took this on. And what he did was he examined a natural source database. This is, again, another database that comes from human food. There are a lot of such databases because the food and flavor industry is quite interested in developing better tasting foods for us. And so we take advantage of these data sets. So Tom worked together with our collaborators at the Salk Institute, computational neuroscientist Tanya Sharpie and her student Mendy Xu. And in the last couple of years, Tanya has shown that hyperbolic models capture the underlying structure of natural odor sets quite efficiently. And the intuition for why hyperbolic metrics do a good job at doing this is that all natural data sets appear to have underlying hierarchical structure that is well captured by these types of hyperbolic geometries. And so the idea is that the hyperbolic, embedding these kinds of data sets in hyperbolic models provides a continuous approximation of the tree distances in these types of hierarchical networks. So for instance, two odors, for instance, like B5 and B6, that might have quite a short distance in a Euclidean embedding, when they are embedded into a hyperbolic model, would have a longer distance that reflects the fact that their most recent common predecessor is farther up in the tree. Okay, so we work together with Tanya to use these types of metrics to quantify relationships of odors in these natural odor source data sets. And we asked whether now, having these two types of descriptors, how these compared against distant neural distances that were measured in the input space, in the mushroom body space, as well as in the modeled Kenyan cell space. All right, so here is what we saw. Here is the, this is similarity of relationships of odors as measured by the OR representation in the input space. What we see is that OR distances are modestly correlated with the chemical similarity of the odors, but not really so much with their natural source distances. Whereas when we look at now data that comes from Remy's studies of looking at neural distances in the mushroom body, we find that distances between odors as measured in the mushroom body space are not particularly well correlated with chemical similarity, but in fact are correlated with their natural source relationships. Okay, and we can kind of summarize this up here, which shows that OR distances are better correlated with chemical descriptors rather than natural source distances, and vice versa for Kenyan cells. And of course, these modeled Kenyan cells, right, which maintain the input relationships from the OR, of course, look more like the OR relationships in this case. All right, so these relationships in this natural source space that we've been quantifying, we think that they arise due to these underlying these metabolic pathways that are generating these dependencies among the odors that are in the natural sources. And so we thought to examine this by looking at additional data sets that were available, including this Metapsych database, which is a highly curated metabolic pathway database that comes from many organisms. And I'll note that, of course, these are large, diverse data sets. Some of them may not be necessarily relevant or interact with Drosophila in the wild, but this is sort of the types of data that are available to us to work with, as well as this natural products database that is more similar to this food database that we have already worked with. Okay, so as an example, right, a class of odorants that are particularly important to plant-animal interactions in the world are terpenoids. So this GPP, this geranyl diphosphate, this intermediate in terpenoid biosynthesis, is the precursor to a large number of these monoterpenoids that, monoterpenes that are a major class of volatiles that are emitted by plants, and they have a relatively high vapor pressure, so tend to be volatilized and can act at a distance as attractors or deterrents to different animals, for instance, for attracting pollinators or deterring herbivores. So we generated another odor panel that is enriched for these plant volatiles. And you can kind of see now here, this is now clustered by chemistry. You can see that it now has a more limited range of chemical diversity, but is enriched for these odors that we think might be relevant in natural pathways for Drosophila. And so now we can compare the chemical and metabolic odor relationships. These are distances between these odors now measured in these Metapsych or Lotus natural product databases. And when we compare these against their distances in the original food database that we've been working with so far, this VCF food database, you can see that gratifyingly, some of these different metrics that we think are related to, should be related to one another, indeed look quite similar. And now we can compare these relationships to what we measure in the fruit fly in both the input space and the ORNs, as well as in the Kenyan cells. And so what I hope you can see is that the Kenyan cells do capture some of the odor relationships that we see more in these metabolic pathways, in particular these pathways with beta-myrcene and several of these other monoterpene compounds downstream of this GPP pathway. And you can see in this case, one interesting note about this panel is that there seems to be a weak relationship between chemistry and natural source and natural source relationships. And we actually think this is something we're interested in studying more broadly, whether this is specific to this particular, to the more natural products data set, or more generally across odor space, whether there is this systematic relationship between chemistry and metabolism that shows up. Okay, so as kind of a general outlook, and to sum up here, we're really interested in trying to understand what are the sources of structure in the olfactory circuit, right? Where does this structure come from? Where is it built in? Can it be modified by the animal's experience or its environment? And so we have developed tools that allow us to track representations now systematically across each step in this pathway, from the ORN axons, which I've shown you a bit, to the dendrites of the second order neurons, to the output boutons of these PNs, and then again now back up to the somata. And we can trace these relationships successively at different stages of processing, and you can see the successive decorrelation of odor relationships as you move through this circuit. But of course, this major reorganization that we see seems to occur at this last stage, somewhere between where it receives output from the earlier stages of processing at the PN boutons and the emergence of the representation at the cell bodies. And so we're quite actively studying the mechanisms that are in place in the mushroom body that could be mediating these transformations. So I just want to sum up here, right? The classical framework is that randomly connected networks such as the mushroom body, like these cerebellum-like circuits, like the mushroom body, would be expected to maintain their input relationships in this high-dimensional representational space. What we actually observe is that we see a reorganization of odor relationships that diverge from what we see in the input space. And what this tells us is that the mushroom body is not decorrelating equivalently in all directions. Okay, and that odor relationships in the Kenyan cell space appear to be better predicted by their natural source relationships compared to the chemical relationships of the odors, which perhaps appear to be better captured at earlier stages of processing. And what we think that this might be good for, right, is that odors their representations may be being remapped to allow them to be more efficiently generalized to other odors that are more likely to co-occur with those odors in natural sources that are behaviorally important to the animal. So for instance, you might want to remap this alcohol and ester together because they tend to be a signature of spoiled fruit versus this alcohol and ester, which might be a signature of ripe fruit, which would allow you to more efficiently categorize stimuli, in particular noisy stimuli, in the real world as being of the category that you want to eat or that you want to avoid, for instance. And I'll just also note, finally, that these data are kind of particularly interesting in the context of emerging results that are coming out of cerebellum that show that representations in cerebellum actually are denser and less decorrelated than you might expect, as well as connectomic studies that are coming from cerebellum and from fruit fly that are showing unexpected structure in the inputs into these circuits. So I'll just leave you with this, that in addition to these important organizational axes in the physical chemical space that allow us to predict how odors may smell, we suggest that there are additional axes that we should be paying attention to, these axes of how odors are organized in the natural world, that will give us additional ability to able to predict how odors drive perception and behavior. And I just want to thank the people who've done this work. This is my group. It is my greatest blessing in life to be able to work with them every day. They are fantastic. And I also want to acknowledge this network that I'm part of, the Odor to Action Network that was funded by the NSF. And in particular, Tanya Sharpie and Brian Smith, who worked on the natural odors space project together with us, as well as funding sources. Thank you so much.
Thanks so much, and just a reminder to come up to the mic to everyone who has a question. I had a quick question initially. I'm wondering what you think about what you would expect in different species with different diets, or perhaps different early life experiences.
Yeah, absolutely. I think that's a really interesting question. I think the most provocative idea out there, and I'm just tossing this out there, I don't fully believe this, but is the idea that maybe there is actually a conserved olfactory perceptual space across all or lots of organisms, right? Because we all live in this common world that is shaped by these common metabolic pathways. I can smell a rose like a fly can smell a rose. Now, I guess the question is, should the odors mean the same thing, and will our individual brain, right, will each species' brains remap these odors in accordance with the relationships that are most meaningful to them? So that's where I think there will be divergence. But I didn't show this because I feel like it's still kind of preliminary, but we do have preliminary evidence that human perceptual distances look much more like Kenyan cell distances than ORN distances, which is kind of a hint in this direction that there is this kind of core common perceptual space that we all operate in. But obviously there are going to be very individual particular chemicals signals that mean something very specific for each species. Yeah.
That's super cool. I'll take a question here.
Thank you. Absolutely beautiful talk. I'm curious, I guess you've already alluded to this, but in the natural food sources, you're feeding them in the fly facility. I guess some of these metabolic pathways are ongoing. And you also highlighted another possible source of bias, which would be the innate odor processing. And I know that at least in humans, I think some other animals, we have, early animals have some positive negative valence ordering in the odor space. And I'm curious whether there's already some evidence for how much of this can be nature versus nurture, and what your thoughts are.
Yeah, absolutely. I have no doubt that a lot of this is developmentally specified. So, evolutionary processes have shaped these developmental programs that build in structure into the circuits that we are reading out. So where that structure arises in the mushroom body network, I think is really interesting. So I would love to do, when we began, we wanted to do these experiments in the lateral horn, because that is the obvious, since that is responsible for innate processing, you would think that these types of relationships would be much more obvious and show up, more readily in that circuit. The problem is that the genetic tools that allow you to do population-wide analysis of the representation are quite limited there. So we started where the tools allowed us to work as well. But one possibility is that these relationships are structured in the lateral horn, and that the lateral horn could somehow be training this arm of the mushroom body through mechanisms that are still unknown, right? And that provide this kind of basic substrate that then perhaps experience can act upon. And we are very, very actively pursuing the experiment where we try to raise animals in environments that contain odor relationships that never would occur in nature, as far as we can tell looking at these databases, and trying to see if we can remap or alter the odor space in that way. Yeah, absolutely, great question.
We have one final question, and could the next speaker come up and set up the computer?
Yeah.
Go ahead.
Hi. Beautiful talk. I think this is a conversation because you asked already the question I want to ask. Then my question is, do you speculate that the weight matrix between the ORN and the canyon cell not to be random to start with? Because that's a hard constraint you start with and not seeing the effect. And the second, on the same line, is there a for animals that are like mice, there's also a lot of plasticity going on. Do you think there is a spectrum of innateness and also plasticity that's differ between different kind of animals that needs different level of plasticity to happen?
Absolutely. Yeah, these are great questions. The answer to the first question, I didn't really want to get into this, but this has been a very active area of research that the connectome has weighed in heavily on, which is whether or not there is structure in the connectivity between PNs and canyon cells. There is indeed structure. There is structure both in the sense of some glomeruli have more inputs than others, so there's a frequency bias across the glomeruli. And then there's also structure in the sense of overconvergence. So certain combinations of glomeruli connect to downstream canyon cells at a higher probability than would be expected just based on their frequency. Okay, so there's this overconvergence tends to be among glomeruli that respond to food-like odors. So this is a possible metric. I will say that when we implement those connectivity biases into our very simple-minded model, it does not produce enough transformation that matches what we actually observe experimentally. So there's still something that is missing that we are very actively trying to track down. We feel like we can measure almost all the different parts of this circuit, and so it is inevitable we will find it eventually. But yeah, the second question, I think maybe can we talk about this offline, just because for time I don't want to cut into the next speakers.
Thanks so much. So let's give a round of applause. Thank you to Betty Han, and let's welcome our next speaker, Khue Tran.
Hi everyone, my name is Khue. I'm from the Koulakov lab at Cold Spring Harbor Laboratory, and today I'm excited to talk to you about our recent project that is weight transfer in the reinforcement learning model of songbird learning. So we're interested in the songbird system because it provides a fundamental example of sensory motor learning by repeated practicing with trial and error. It's been shown in experiments for a long time now that male juvenile songbirds will learn to reproduce a tutor song. This tutor song could be coming from a nearby male adult, and this is a process that happens rapidly and accurately. So we can imagine this is the tutor song that is to be learned. The song is represented in a spectrogram, which shows frequencies on the vertical and time on the horizontal. Prior to learning, a juvenile songbird will look something like this, as shown in this spectrogram. So we see that there's quite a large difference between the juvenile song and the tutor song. But just 40 days later, through a process called vocal babbling, where the bird practices the song many different times, each time making slight adjustments to the song so that every iterative song is better than the previous. After 40 days, the adult song looks something like this. So the adult spectrogram now much more resembles the tutor song compared to the juvenile song just 40 days ago. So this song is now considered learned or crystallized and will be maintained throughout adulthood for the bird. So we're interested in this behavior, and we want to see how the underlying circuit of the songbird system can give rise to this learning. So our model is based off of the songbird circuit, which is shown here in this picture. And our model combines two main pathways to song learning. The first pathway is the song production pathway. So this pathway starts from an area called HVC. HVC then connects to the region RA, which has downstream connections to motor neurons to then produce two output traces for the model, one trace for amplitude and one trace for pitch. With these two outputs, we can reconstruct a song. And the second part of our model involves the song learning pathway. So this pathway is crucial because it receives direct VTA inputs into the region area X. Area X also receives inputs from HVC. And area X forms a feedback loop with LMAN, which then connects with RA, which connects our song learning pathway and our song production pathway. So I will start with HVC, which is a region that has activity to define timing context within song. So looking at a song spectrogram, we can look at what HVC activity looks like during the time course of the song. And what we see is that HVC neurons activity shows very stereotyped, predictable activity where specific neurons will only be active for that point in song. So this is important because it provides our entire model and the bird the context of where they are in the song. And this is how we have chosen to represent HVC activity in our model. The next region we're interested in is L-man. So I mentioned before that the bird is able to learn the song by a process called vocal babbling, which requires the bird to generate different songs every time with slight variations. So the source of this variation has been largely attributed to the region L-man. So we see that from a regular song spectrogram, with L-man to be silenced, the song then loses all variability and becomes extremely stereotyped. So this is detrimental to learning because then the bird is not able to improve upon the song. The next region that is important is VTA, and we all know that VTA carries dopaminergic signals, in this case from the auditory cortex, that carries reward prediction error in the form of reward to the region area X, and then which makes area X important because it is the most important region for song learning. It not only receives connections from VTA, as I mentioned, but also HVC and L-man. So in experiments, they see that if they were to inactivate area X in juvenile birds prior to learning, song learning is not possible. However, if this happens after learning, when the song is already crystallized, the song can still be produced. So these pieces of evidence points us to a potential hypothesis that is the synapse between HVC and area X, that is the synapse of song learning. Previously, people have studied the system, and they consider reinforcement learning models for this system. So here in 1993, Doyon Sinofsky suggested this model where they modeled the song production pathway, essentially, which starts from HVC, which then connects through region RA through weight matrix W. So these are the weights that will be learned. RA then has downstream connections to motor neurons to produce the song, and they were able to show that with this model architecture, using a learning algorithm known as weight perturbation, an algorithm that requires two main components, one component for a gradient estimate and one component for perturbations, both of which have been shown to exist in the songbird system, namely the reward signal from VTA for a gradient estimate and the L-man as a source of exploratory noise for perturbations. So by training this model using the learning rule, they were able to show that you can learn a song over trials, and you can see here that accuracy essentially plateaus by learning the song. More recently, the model has been updated by Fiat and others to incorporate a learning rule known as node perturbation instead of weight perturbation. And in this paper, they were also able to show that by implementing this model using that learning rule, you can learn a song, and we see that with the decreasing errors. So what we want to do in our model is to reconcile the experimental findings that seems to suggest song learning actually occurs in this pathway, the song learning pathway, while being consistent with the known circuit, which contains two distinct pathways. So what we propose is a model with two stages of learning for the two different pathways. The first stage is reinforcement learning, but at the synapses between HVC and area X. So this is the main difference that we are making compared to the previous models by making these weights M here, the weights that are being updated to with reinforcement learning. So we propose our reinforcement learning update rule is a product of these three elements. The first element is the activity of L-man which produces exploratory noise. The second element is the activity of HVC, which provides timing context. And then we also have reinforcement reward from the input from VTA to area X. So by training just this network with following these arrows in red here with this learning rule, we can train this model to learn the song, but this doesn't fully complete the picture because then we're not explaining anything that is happening in the song production pathway. So the question is, how's the learned information that is in this pathway end up in this pathway? Because we know that this is stable throughout adulthood. So then we hypothesize that there's a weight transfer, pictured in this blue arrow here, from weight matrix M to weight matrix W. So there's a weight transfer between the synapses, between HVC to area X and HVC to RA. And we further propose that this weight transfer takes place via an STDP mechanism. And so the weight transfer, the weight update rule that we propose is this, which is a convolution between these three elements. The first element is the postsynaptic activity of area RA. The second element is a kernel to maintain copying of the weights, and the third element is activity of HVC, once again, for timing context. And we arrived at this weight update rule because we see by the pathway here that HVC, which goes through area X, comes through a pathway via the thalamus to eventually end up in RA. So we can roughly estimate the activity of X with the activity of R. And we know that the activity of X is exactly weight matrix M here times HVC activity H. So we can rewrite the formula to look something like this. And since we propose that there's a weight transfer, we want the transformation from the M weight to the W weight to essentially be the identity matrix, which constrains our kernel here, and we can solve for the kernel and ends up looking something like that, which is known as the symmetric form of SCDP. So by training this network using the two learning rules I mentioned, we find we can train our network to learn a very simple song. And what we see from those results is that firstly, we see the correlations between these two weights increase over iterations of training. We also see that the model can eventually learn a song by seeing the saturating reward. And the reward here is a measure of how well the song that is produced matches to the tutor song. We can also visualize what the weights look like prior and after training. So before they were both randomly initialized, but after training we see that they are quite highly correlated with each other, which confirms the weight transfer. And we can also plot what the output traces of our model looks like. So there's one trace for amplitude and one trace for pitch, and you can see how they correspond to the target values. The more exciting part of this model, however, is that our results are able to explain experimental findings that previously was not able to. So here we're looking at the reward after training of producing the song by implementing the previous model. So this is the model that essentially combined the song learning and production pathway. We get performance that's very close to being perfect. When we implement our model, which separates the two pathways by implementing two different learning rules, we do lose a little bit of performance. But what we lose in this performance, we're able to explain in terms of the circuit. So we require that weight transfer needs SCDP mechanism to be to successfully transfer the weights. Without SCDP, we cannot successfully learn the song. Also consistent with experiments, if we were to inactivate LMAN, which is the source of noise, we also cannot learn the song. More crucially, as I mentioned, the role of area X in juvenile song development. If area X were to be inactivated during learning, we see that the song cannot be learned. This is similar to what they saw in experiments. However, because of the weight transfer, if we were to inactivate area X after learning, we see that the song can be recovered. So we're very excited about these results and this style of computations, and we want to see how this similar framework can translate to other circuits. One such example is human speech acquisition circuit. So this paper highlights a high homology between different regions in the bird circuit and human circuit. So we see here that HVC and LMC and LMAN and Broca's area seem to follow a similar evolutionary track. So in conclusion, we have two main conclusions. The first one is that based on the model that we have proposed that separates the song learning and song production pathway using two different learning rules, we require that the weight transfer from the song learning to the song production pathway requires a specific form of SCDP that occurs between HVC and RA neurons. And our model can also replicate results previously seen in experiments, such as the results of inactivation in area X. And that's all I have. Thank you for your attention.
This is super amazing, really, really beautiful. Just, that very last slide that you put up suggested homology with the primate. Which weights will be transferred between which circuits in that, if that homology were true? Just because my mapping wasn't fast enough.
Yeah, that is the future direction of what we're currently working towards. We've only shown that this works in the songbird system, but if I were to hypothesize from just looking at from that picture, we would see that there's a similar set of weights from HVC to area X, so that's LMC to this region that I don't know the name of.
Okay, I'll speak later.
So maybe it's related, but what do you assume on the connectivity from area X to the RA? Do you need to relay the activity of area X to RA?
Yeah, so I think it's been shown in experiments that there is an indirect pathway from area X to RA that is connected through a region through the thalamus. It goes then through LMAN, and then LMAN goes to RA. So the signal from area X to RA is not a direct one. It is disturbed by noise in the middle. But we propose that there is a mechanism, potentially there is a mechanism that can facilitate the weight transfer.
Okay.
While the next speaker is getting up, I have one final question. I'm just wondering, I know there's been evidence of sleep replay in this system, and I'm wondering if you think the weight transfer is happening during song learning itself, or is happening during sleep or some other time?
Yeah, so we didn't explicitly model this in our model, but I think in experiments they have shown that the consolidation process happens over multiple days. It's not just like a one-night thing. So I think it could be possible that this is happening at nighttime because it happens over like many days.
Let's thank Khue Tran again and welcome our next speaker, Rishika Mohanta.
Hello everyone, I'm Rishika Mohanta, and today I'll be speaking about work I did over the last 2 years at the Turner Lab in HHMI Janelia Research Campus. As we go about our daily lives, we are often faced with situations where we have to make decisions despite uncertainty and not knowing if the world is suddenly going to change. And we might not be able to predict that. For example, I recently moved to New York, and I've been looking for a good place to get some nice bubble tea after a day at work. And so I started visiting the 2 nearest tea shops, and initially I figured out that Kung Fu tea is where I am most satisfied. But over time, I noticed that the supplier changed, and I got some new specials at Boba Whale, so I had to change my preferences to deal with the consequences of the world changing. This, in fact, is in essence just a reinforcement learning task called the two-arm bandit, but in our real life. Now, how do the circuit in our brains implement a decision like this in a situation where I have to constantly update my preferences and change my behavior? To attempt to get a mechanistic understanding of the circuit principles underlying this behavior, our lab decided to study the two-arm bandit task in a highly tractable model system, the fruit fly. To do that, Aditya Rajgopalan, a grad student in the lab at the time, built a 2 alternative choice setup where single fruit flies could walk around in a Y maze with 2 odorized arms here in green and orange. In this video, you will see an example trial. The fly starts in an empty arm with no odor, marked in white, and only when it makes a decision by walking upwind to the very end of an arm does it receive a reward marked by the red flash, which is a stimulation of the optogenetic sugar sensory neurons. At which point, after it makes a choice, the trial ends and we move on to the next one. Using this setup, he was able to show that flies behave surprisingly similar to other animals, including primates. In these plots, you can see that the choices made by the fly are in blue, and the reward probability across the session is in white. And as the reward ratio changes over the session, the fly and the monkeys change their reward ratios, choice ratios to match the reward ratios. Based on this result, known as the matching law, Aditya was able to show that flies use reward expectation to generate this matching behavior. But is the reward that we expect from the world around us and all that matters when it comes to decision making? Is there something more? To answer that question, I built a high-throughput version of the same rig, which would allow us to look at 16 flies simultaneously in independent choice tasks. And with some elbow grease in a really short time, we were able to collect a large data set of 360 flies with hundreds of trials per fly. Then, to analyze the data, we looked at the rich history of cognitive science and found that there were many different models to understand how the behavior is happening. So we fit these models to that and then asked, what do the best models do? What do they have in common? So let's start with the best regression model. The regression model allows us to look at how the choices made, the rewards, and the association of the choices with rewards have an impact on it based on different points of times in its past. When we do that for the fly, as expected, we see the strongest effect in purple is a recency-weighted integration of rewarded choices. This, in a sense, is nothing but a reward expectation. But as we expected, there was something more. We saw that the most recent choices that the fly makes biases the next choice that it's going to make. It has a tendency to keep repeating what it did last time. Surprisingly, this was very similar to a recent result in mice from the Sabatini lab at Harvard in a very similar experiment. Further, this result was not even limited to just a regression approach. Let's say we look at the reinforcement learning model of this behavior. Now, in this plot, I am going to plot in a 2-dimensional space how the reward expected for the chosen odor changes depending on whether it's rewarded or not. Let's say we start at an arbitrary state with 1 being perfect expectation that you are going to get a reward, and 0 being no expectation that you're going to get a reward. Starting from a random point, the model is going to describe how it's going to be updated based on the reward contingency. We would expect that if it is rewarded, it is going to saturate towards 1, with expecting reward for the chosen order, and in an absence of reward, decay down to 0. And that's what we were expecting, that in the first case, where it's rewarded, we'll have the black cross on the left plot, and when it's not rewarded, we have the black cross on the right plot. But we observe something different in the fly. We notice that even when the fly makes a choice that is not rewarded, the reward expected of the chosen order approaches a small positive value, which implies that even when the fly is not rewarded, it still maintains a preference towards that last chosen reward. This shift might appear to be small, but it has a big impact as you integrate over multiple trials, and there's a qualitative difference between 0 and a small positive value. Therefore, based on what we observed here, we define the behavior that we observe as perseverance or a drive to repeat previous choices. In the example that I gave earlier, it would mean I would continue to go to boba whale even if I stop being rewarded in hopes of a future reward. But how does the fly generate this persevative behavior? As many in this conference have shown, the beauty of working with the fly is we have a lot of genetic tools, we have connectomics, and we can try and answer this specific question. And thanks to Betty Hong, she gave us a good introduction into the mushroom body, which is a major neuropil in the fly brain that is involved in what we call experience-dependent value coding. In essence, what it does is it takes the odor identity and combines it with reinforcement information that it gets from its environment to create a readout via dopamine-dependent synaptic plasticity, which in this case is how good or how bad the odor is, the value. This value-generating population then transforms this to guide the actual choices that the animal makes. But what about perseverance? How do we get from this to perseverance? We hypothesize that if the making a choice itself is intrinsically rewarding to the animal, we might see perseverance to emerge as a behavior that is just a consequence of this intrinsic reward. So we hypothesize that there might be a positive feedback loop that takes you from the choice-generating populations back to the positive reinforcement neurons. And that could be the reason we see perseverance. So the first step to doing something like that would be to identify such a feedback pathway. And to do that, we need to know what is generating these choices. In our task, the main thing that guides the choice is upwind walking. So we looked through the data that we have about the fly based on past research, and we identified and focused on this downstream population of neurons known as these upwind neurons. Upwind neurons are called so because of their remarkable ability that when you optogenically activate these neurons, all flies start doing, as you can see in the video, with the red being the optogenic activation, they start orienting upwind, upwards towards where the wind source is, and go straight up. Further, what has been shown is if you ablate these neurons, you lose choice behavior, and you can't learn any order preferences. So this was our choice-generating population. So we looked at the hemibrain fly collectome, and luckily we saw there is indeed an indirect feedback pathway that takes you from these neurons back to the positive reinforcement population, which was great. And when we look at the neurotransmitter predictions, they all seem to be positive feedback. We're like, okay, this might be it. But how do we know that that's actually true? There is an actual functional feedback. We turned back to optogenetics. By stimulating this downstream population of neurons, we were able to put the fly on a ball where we can give it closed-loop wind and odor stimulus. And when we did that, and we paired the activation of these downstream neurons with an odor, over successive trials, we saw that as you train the animal responds by going upwind in response to the odor itself without even stimulating this. So in subsequent trials, the fly has a positive feedback to keep going upwards. This, for us, confirmed an existing positive feedback pathway. But even if we have this, how do we know that this could generate perseverance? To validate that, we went back to in silico circuit modeling, and we took the pathway that we had identified in the fly and converted it into a reduced model that takes into account the circuit architecture, the plasticity rules that we know at these output synapses, and combines all of them to generate an upwind drive. And then using this upwind drive, we were able to fit it to the behavioral data from our experiments. and then look at these models and give them a chance to do the exact same experiments as the fly. We can take that behavior and compare them using the logistic regression coefficients. And surprisingly, what we see, if you look at the second one in the green, the fly does manage to create a perseverance, like the model appears to create a perseverance-like signal just like the fly, with a small difference that the time scales seem to be longer. This is something we expected because in the fly, there are actually multiple time scales of plasticity, and our model assumes a single one. And then it becomes a little hard to disentangle what's choice and what is reward association. So the most interesting thing, however, with this was when we ablated that feedback, we were able to completely get rid of this perseverance without affecting the reward association at all. We are right now, so with this, we have shown that flies show some sort of a perseverance behavior in a 2-arm banded task. Using genetic tools and connectomics, we have shown that there is a potential intrinsic reward pathway that implements this perseverance. And using a connectome-inspired model, we've shown that it is sufficient for generating this perseverance behavior. Currently, we are in the final progress of validating the role of these upwind neurons and the feedback for generating this behavior in vivo by using targeted genetic ablation and calcium imaging. Further, we have an ongoing collaboration with Google DeepMind to use some novel interpretable deep learning tools to go beyond value and perseverance and ask what else could be there in the behavior. And finally, we have a poster with the Anne Hermanstad lab at Janelia, which some of you might have already seen, investigating how we can design highly informative tasks to give to flies and potentially also humans and see how they can learn short-term rules that guide the world. This work was made possible by the work of a lot of people, and I'd like to thank all of them. Glenn Turner, particularly my advisor, and Aparna Dev, the technician who collected a large amount of the data that you see today, and all our collaborators who have been helping us push the boundaries of what we think about decision-making and economic choice in flies. And if you have any questions, I'm happy to take them now, or you can catch me later in the conference or at the workshop.
Awesome talk. Reminder to come up to the mic with questions. I have a question. And so the title of this session is Sensing and Learning, and I think it's a really cool pathway that you've noticed kind of back from the learning to the sensing neurons. And I wanted to get your take on kind of other things other than perseverance, or maybe other species that might have similar kind of principles.
I think the general thing that we observe over here is a very simple thing, that our reinforcement neurons are not just doing the sensing. The sensing is happening at the sugar sensing neurons. The reinforcement neurons, just which is pretty close to where the sensing is happening, are already doing a lot more. So in the first part of the talk, I talked about reward expectation. We actually show there is a potential reward expectation error signal that is guiding that's happening at the same one. Now it's getting more feedback down from cognitive things. The general principle is the output of these systems don't really disappear. They have response on to what's happening further away in time. I think the general idea is we need to also think about long-term consequences of learning that are going to shape the way you see the world. So reinforcement fundamentally means something else because of your past experience.
Cool, thanks. Go ahead.
Hi, thanks for this work. Thanks for the very nice talk. So I was just wondering, is another interpretation of your perseverance prior on how quickly the reward contingencies change? And if so, is the implementation of that where you would expect it to show up in the circuit?
That is a very, very interesting point. Now, the question is it something that it very much could be a way for the animal to deal with that? Our experiments sampled a wide variety of volatilities of the environment, and we didn't see any strong changes on the perseverance with respect to that volatility. However, it could be that the fly has adapted to a certain level of volatility in the world. The reason why we think this might be important is that either in a volatile world, you need to keep going. There might be dangers of switching to something new, so you have a tendency to keep going. One even more naturalistic situation where that might be important is you're walking in a plume that you know might give you a reward, and suddenly it disappears. It is more likely than not that if you keep going ahead, your odor source might be up there, but if you give up immediately, then you might not be able to behave well or perform well or survive in the natural world. So choices over here, or in a more naturalistic bloom context, I think that's where this becomes very relevant.
Great. Thank you.
And one more question, and can the next speaker please come up and get set up?
Thanks, that was a great talk. My questions were actually partly the previous questions also, but I think I can extend it a little bit. I'm curious whether you think changes in the past experiences of the flies could also contribute. It's similar to volatility, but I'm like, are the flies hungry or not hungry, and would that change their perseverance? And maybe related, is it perseverance or memory? Have they forgotten that they went to the previous arm?
I think that's a great question. There's definitely an internal state dependence. I can't say too much about what we're exploring because that's a part of the collaboration with DeepMind. But there is a starvation state dependence, and we can see that pretty clearly. Like even not just for perseverance, just even reward learning rates change, and there's a lot of things. Motivational state is an axis that completely changes the scale of behavior we see.
Got it, thanks.
Thanks. Let's thank Rishika Mohanta and welcome the next speaker, Denis Turcu.
Hello, and thank you for the opportunity to present my work. I'm Denis Turcu, and I'll tell you about computational insights from weakly electric fish. These fish have an electric organ in their tail which generates small and brief electric pulses in their environment. They are equipped with an array of electroreceptors in their skin tuned to detect minute field perturbations due to nearby objects. Live objects generate slightly different perturbations than inanimate objects due to their capacitive properties. And the fish process this information in a specialized brain area called the electrosensory lobe to characterize and detect these nearby objects. They can identify both spatial and electric properties of these objects, and we are going to look at how they can characterize and detect these objects. But first, why should we study such an unusual sensory system? As you've seen already, it's a fantastic system, and also it provides us broader insights into computational principles. In particular, it mimics computational challenges from other sensory modalities, notably from vision. And it also introduces new challenges due to the sharp decay of signal with distance, 1 over distance to the 4th, because the fish emit 1 over distance in their vicinity, and the object perturbation gives back a dipole field of 1 over distance cubed. Now, scale is a problem in other sensory systems as well. For example, in vision, we can identify objects regardless of how far they are, but the sharp decay, this dramatic decay in the electrosense can reveal new and elegant solutions to this old problem. So I'm going to model this system end-to-end from sensory information all the way to object identity, and I'm going to do so abstractly using convolutional neural networks. I'm going to present a comprehensive analysis of this system, which involves multiple different components. I'll start with a physics model that I built to generate sensory data. And then I also built an electroreceptor model based on LFP data that processes the sensory data and feeds into the CNNs. Oh, sorry. And then I'll present a few computations that help the CNNs actually perform active electrolocation. So diving into the physics model, this builds on previous work, is 3D and analytic. And it's reliable and fast, which makes it reliable and fast to generate necessary data for neural network training. I'm showing here equipotential surfaces around the fish and a nearby object which produces an electric image on the skin. Importantly, this model allows us to look at both both electric properties of nearby objects, the resistance and the capacitance, because it is dynamic, which is something that previous models did not fully capture. We are testing this model in freely swimming fish. These charges emitted by the fish are recorded here by 10 pairs of electrodes, and we found that the model traces this data very well. Moving on to the electroreceptor model, these electroreceptors process the sensory data directly, so we built a model to be able to study foraging. This model consists of 2 sensory cells which project directly to 2 somatotopic maps, and perturbed discharges stimulate those cells, and we recorded LFP during stimulus presentation. We then built a model, an electroreceptor model based on these recordings. And I'm asking here just what is this model useful for? So looking at variability of the stimuli, we found that these 2 principal components explained almost all of the stimulus variance, which nicely matches the number of sensory cells available as well. The first 2 principal components show that stimuli variability comes from changing the resistance and the capacitance of the nearby objects. And in particular, in this space, delicious worms are distinguishable from inanimate objects, but also distinguishable from the broccoli equivalent of worms. We found that the electroreceptor model modulation space is merely a simple transformation of this PC space, meaning that the electroreceptors are tuned to detect these electric properties of the nearby objects, while the responses of the 2 cells are slightly correlated, possibly to allow for error correction. So we investigated how could this model serve for behavior. We built a neural network, small neural network, to extract electric properties out of the model's responses. And we found that these networks could reliably and successfully extract both capacitance and resistance of the underlying object. Now, machine learning models have not been used in this sensory system, but this is a really good system to do so, because now I can simulate the sensory data using the physics model, and I can use the electroreceptor model to parse this sensory information, which allows me to dive into downstream processing of more complex tasks, such as characterizing and detecting nearby objects. And I'll start with characterization. So swimming fish come close to a worm, which generates electric images on their skin. The fish wants to find the location of the worm as well as its size, and the fish wants to know this is a delicious worm indeed, as opposed to an inanimate object or a dull worm. And these fish can perform this task in the presence of noise and in total darkness. So we modeled this characterization problem via convolutional neural networks and found that these networks had a hard time solving the full task very well, with especially high errors for the electric properties. But we saw that other models could extract these properties when the spatial properties were fixed. So we restricted the task to spatial properties only and found that now these networks perform very well in this scenario. So we went back to the electroreceptor model modulation space to investigate the performance issue. This feature space corresponds to the responses of the most modulated receptor for an object that is close to the fish. When the object gets farther away, this feature space shrinks, and it shrinks rapidly by a degree 4 polynomial. This results in the CNNs needing to compute across many orders of magnitude and perform multiplication, which is very hard, partly due to bounded nonlinearities and mostly due to the multiplicative operation. So to solve this performance issue, we provided the CNNs with a hint. We informed them that they should use the distance they find to learn a multiplicative rule which scales the feature space based on distance. Downstream networks learn this multiplicative rule and then successfully extract the electric properties of the nearby objects. This suggests that extracting all object properties independently is hard because they interact, but using some physics insights to loosen the interactions helps a lot in this case. And also this provides, this sets a hypothesis for experiments in higher electrosensory processing stages, which can examine how this computation could be implemented neurally. Besides characterization, another part of foraging is detection. So in previous experiments, freely swimming fish were trained to detect an object in the tank. They were asked to choose the correct partition where the object was placed before crossing the decision line. Behaviorally, they could detect these objects when they were 7 centimeters away from the decision line, but in individual neurons, the detection signal vanished beyond 1 centimeter. We wanted to explain this computationally. So we generated electric image data and input enough noise such that the single receptor neural network performance matches the single neuron detection threshold. We wanted to know how many receptors would be needed in this case to match the behavioral limit. We were hoping to get a number close to thousands, the number of receptors that fish have. But increasing the number of receptors would barely improve performance. So why is this? The signal decay with distance, 1 over distance to the 4th, combined with the typical square root signal-to-noise scaling, means that we need the number of receptors to scale like distance to the 8th to stabilize performance. We observed this in simulations, but this requires 2 to 3 orders of magnitude more receptors than the fish have on their whole body, which is highly unrealistic. So is there another way in which multiple samples could help? We found that motion coherence is key for this task. Fish swimming, when a fish swims by an object, the electric image moves on their skin accordingly. When the noise is high enough, objects that are far away cannot be detected. But if the fish could use a representation of time and compute the response slope for each receptor, the objects become a lot more easily detectable. So using this slope computation as pre-processing for the neural networks model, we found that indeed they could solve the task and detect the objects remarkably well with very few receptors. Fish could potentially use other strategies as well to solve this task, such as tail motion or parallax, and we're currently examining ways to confirm this slope computation experimentally. Nevertheless, why is this slope computation so helpful, since in fact the slope signal scales even worse with 1 over distance to the 5. As it turns out, it also has favorable variance scaling with number of samples to the 3rd, which means that the number of receptors needed in this case would scale like distance to the 10 over 3, which is much better than distance to the 8th. With that, I'd like to summarize the 2 main computations we identified here, which help solve active electrolocation. First, knowledge of stimulus scaling rule results in performant object characterization using internal feedback. And second, motion coherence fills the gap between single receptor and behavioral limits. And with that, I'd like to thank you for listening for the upcoming questions. Thank Larry, Nate, Abi, and Fede, and the fish for being so electrifying. And I want to end with an evolutionary prediction, which is when these fish will move from water to land, they'll skip walking and start electric sliding.
Cool. Reminder to go up to the microphones for questions. And I have a question as people are stepping up. Have you tested in the model multiple objects? And is that even like ecologically something the fish might encounter, is like multiple objects close enough to them that they might contaminate the signals?
Yeah, so I mean objects, looking at the effects of objects at the same time. I have not looked at that yet. That's part of the next steps of the project. The fish do encounter that. What I studied is individual small objects and large boundaries, such as the side of a tank, for example. And sometimes that can, depending on the positioning there, that can enhance the signal, so help the fish, or can interfere as well, depending on the geometry of the problem.
So it kind of reflects it.
It's kind of a reflection, which can help.
Cool.
Oh, yeah.
Hi, thank you. This is wonderful. I have a simple question in the sense that can these fish identify other fish? And if there are other fish, I mean electric fish nearby, do they subtract their signal somehow?
Yes, they are very social fish, which is actually a new project I'll work on. They communicate a lot with these electrosense as well. They use basically their friends' discharges as well to identify nearby objects, which is very cool system. And also they have a separate set of electroreceptors kind of tuned just for communication itself, so they know when their friends talk to them, basically.
Cool. I had one other question, just maybe a little bit more open-ended, wanting to, and I guess while I'm asking this, if the next speaker could please come up and get the computer set up. I was wondering if you could speak to the connection between this system and other like cerebellar-like systems in other organisms.
Yeah, definitely there's a lot of cancellation that was just mentioned that happens in the system, cancellation of the effects of self-motion, for example. So when the fish moves their tail, the whole electric sensory scene changes. Also, when a boundary is present, like the bottom of the tank, the whole sensory scene changes as well. In that sense, basically, they can use that. They can use proprioceptive information to cancel the effects of their own tail motion. And sometimes they even bring their tails, for example, closer to an object, which enhances the signal as well. So that's another mechanism in which they could explore the environment and enhance their signal. Yeah.
Very cool. Thanks so much. Let's thank Denis Turcu one more time and welcome Roxana Zeraati to the stage.
Hi everyone, thanks a lot to the organizers for this opportunity. I'm a graduate student in Levina lab, and today I'm going to present our work on how different learning curriculum would give rise to distinct mechanisms for performing long memory tasks. In order to survive, thanks. So I hope you can hear me now. So our brain needs to perform computations and behaviors over a broad range of timescales. For example, when we need to perform a working memory or a decision-making task, we need to accumulate information over long timescales. Sometimes we also need to perform very fast reactions in order to survive in the environment. And growing evidence suggests that this broad range of timescales in our behavior can be directly related to the timescales of underlying neural dynamics. One of the common ways of characterizing these timescales is by measuring the decay rate of the neural activity autocorrelation. And this very simple measurement have been related to different types of cognitive processes in our brain. For example, now we know that neural timescales in our brain are hierarchically organized, meaning that neurons in sensory areas have faster timescales compared to the neurons in association areas that are involved in cognitive demanding tasks. At the same time, these timescales are not fixed properties of our brain, and they can adapt to the task requirements. For example, engagement in a working memory or a selective attention task can increase the timescales of neural activity. So now the question is that what types of mechanisms in our brain would give rise to this broad range of timescales in neural dynamics? And generally, there are two candidate groups of mechanisms. The first group are biophysical properties of individual neurons, such as membrane and synaptic time constants. For example, it has been shown that slow and MDA synapses might be relevant for performing working memory and decision-making tasks. In the second group, we have network interactions, and different studies have suggested that both the strength and the structure of the connectivity can shape the timescales of neural activity. And while both these types of mechanisms exist in our brain, our question was that which of these two types of mechanisms are more optimal for performing computations that require long memory. And during my talk, I'm going to show you that, first of all, the answer to this question depends on the learning curriculum, and then I will show you some results supporting that network-mediated timescales might give rise to more stable solutions for solving these tasks. In our study, we use recurrent neural networks that we train them to perform different types of memory tasks, and then we reverse engineer them to understand what types of mechanisms were involved in the computations. So the dynamics of each neuron in our network is given by this equation, which evolves over discrete time steps t, and then for each neuron, we define a time constant parameter that we would train together with recurrent weights. And this formulation of the model then allows us to explicitly study the role of different types of timescales in the computations. So for each neuron, we have a time constant parameter, which gives us a time scale of a neuron in the absence of any network interactions. So you can think of it as an approximation of individual neurons' properties. And it is very similar, for example, to a membrane time constant in a leaky integrate fire model. Then for each neuron, we can also define an effective time scale that we measure from its activity autocorrelation. And this effective time scale represents the time scale of a neuron when it is embedded in the network. So generally, this effective time scales can be a function of recurrent weights and the time constant parameter. And depending on the network structure, it might be equal or larger than the time constant. We train our networks to perform different types of n-back memory tasks, and during all these tasks, the network receives a binary input over time, and it needs to produce a binary output over time. And each input digit is presented to the network for an arbitrary duration of t. For example, in one of the tasks, which is the n parity task, the network needs to compute the binary sum of the last n digits that it observes at each moment of time. So in this task, n defines the memory requirement of the task. So the larger the n, the larger the memory requirement. We train the networks to perform n parity tasks with different values of the n. And what we realize is that these networks can easily perform tasks with a short memory requirements, but they start struggling to learn the task as the n increases. And practically, when the memory requirements reaches to any value above n equal 20, we practically couldn't train these networks to easily perform this task. So what we ask is that, can we come up with a curriculum that would help these networks to perform long memory tasks? Because previous studies have suggested that both the sequence and the composition of the learned task can help the training speed and also changes the internal dynamics. And actually, we came up with two different curricula. So in the first curriculum, which we call the single head curriculum, first, we would train the networks on the simplest possible task, which is the n equal 2 task, and then we gradually increase the task difficulty. So after the network has learned how to perform the n equal 2 task, then we train it to perform the n equal 3, n equal 4, and so on. So this very simple gradual increase in the task difficulty helps these networks to learn relatively long memory tasks. However, these networks suffer from a problem known as catastrophic forgetting, meaning that the network that has been trained to perform a longer memory task can no longer perform a task with a shorter memory. For example, a network that is trained to perform n equal 10 tasks can no longer perform the task for n equal 5, despite being trained for it during the course of the curriculum. So to solve this new problem, then we came up with a second type of curriculum, which we call the multi-head curriculum. And in this curriculum, we also start from the simplest possible task, and we have a gradual increase in the task difficulty. But the difference is that at each step of the curriculum, we add a new readout head for each n. So for example, after the network has learned to perform the task for n equal 2, then we add a new readout head for n equal 3, and then try to train it to simultaneously perform the task for n equal 2 and n equal 3. And in the next step, then we add a new readout head for n equal 4, and so on. So in this way, we are kind of forcing the network not to forget the solution to the previous task that it was being trained for it during the curriculum. So now let's look at if there is a difference between the performance of these two curricula. The first thing that we looked at is the speed of the training. And you can see here that the multi-head networks are much faster in training compared to the single-head networks, because within the same number of epochs, they can reach a higher value of the n. And this might seem a bit counterintuitive because they have a more complicated loss function, but it seems like that having this more complicated loss function is actually helping them to learn faster. Moreover, we can also show that multi-head networks are less sensitive to the perturbations of their parameters after training. So after the network has learned how to perform the task, if we perturb the recurrent weights or the time constant parameters, we see that there would be a less reduction in the accuracy of the multi-head networks compared to the single-head networks. So the next question we asked was that what types of mechanisms in these networks would give rise to these differences in the performance? And for this, we are going to look at the timescales. So first, let's look at the time constant of individual neurons and how it changes over the course of the curriculum. So we can see that for the single-head networks, as they reach to a point that they need to perform a task with relatively long memory, they start increasing their individual neuron time constant with the task memory. Whereas for multi-head networks, there is a reduction in the time constant parameter, and they want to eventually reach the value that we put for the input update timescale, so how long we were presenting each input digit to the network. So these results kind of suggest that single-head networks are using this time constant parameter to adapt to the memory requirement of the task, whereas multi-head networks are only using this parameter to track the fast changes in the input. We can show this more explicitly by fixing the time constant parameter of all the neurons in the network to some certain value and only train the recurrent weights. And we see that for multi-head networks, fixing the time constant value to the input update timescale t would give rise to the same performance as training this parameter, whereas for single-head networks, we do need the training of this parameter for these networks to learn the task, confirming that single-head networks do need this parameter to be adapted to the memory requirements and learn the task. Next, we looked at the effective timescales, which we measure from the autocorrelation decay of each neuron activity. And we see that in both curricula, we have an increase in the effective timescale. But this picture becomes more interesting when we compare it to the single unit timescale that we saw before. Because in single-head networks, we do have an increase in the time constant of individual neurons and the effective timescale. So at least part of the increase in the effective timescale comes from the increase in the time constant parameter. However, in multi-head networks, the only mechanisms that could give rise to slower effective timescales is the recurrent interactions between the neurons, indicating that multi-head networks rely only on the recurrent connectivity between the neurons to develop slow timescales that are needed to perform these long memory tasks. So to summarize, I showed you that depending on the learning curriculum, networks might use single neuron properties or network interactions to develop slow timescales that are needed to perform memory tasks. However, using network interactions might give rise to a faster training speed and finding more robust solutions, which kind of puts these mechanisms as a more optimal mechanisms for performing these tasks. This is also in line with some of our previous findings that we showed that network interactions can be relevant for modulating timescales in the primate brain. We also have other interesting results that I didn't have time to go through them. For example, how these results would extend to GRUs or LSTMs. But if you're interested, you can have a look at our paper that is now accepted to ICLR 2024. And with that, I would like to thank all the people who've been involved in this work, especially Sina Khajehabdollahi, who has co-led this work, our funding sources, and many thanks to you. And I'm happy to take questions.
Really cool work. The question I had is, did you ever try the other way of getting RNN to learn multiple tasks, which is giving it a task identity as another input rather than increasing the number head? And if so, was the fundamental observation similar or different in that?
So at the beginning, we tried to have like a context input that if we want to train a multi-head network, so to do it with the context, and we realized like training the multi-head together is kind of more efficient in the sense of the training, but we didn't look into like if we do the context, what would be the underlying like mechanisms, and it's a good question, so we need to look at it. Yeah.
I had one question about this architecture and how you might imagine kind of multiple brain regions being represented, or different single or multi-headed networks getting connected together.
So I think that's a very good question, and this is the direction we hope to go for, because we have this hierarchy of timescales in our brain, and we might think of that we might need like different brain areas that they would be connected to each other. So in our implementation, you can have just one population that can do everything, but you can think of it that you might have a hierarchy of multi-head networks that each of them has some sort of timescales, because if you look at each individual brain area, you also see a heterogeneity of timescales within each area.
Cool. Well, This is the end of the sensing and learning session. I hope that you all keep sensing and learning together here. A reminder that the next session is starting at 6 o'clock. It's a slightly shorter break. And can we please thank all of our speakers